Before bringing anything, including your arms, into the BSC, be sure to wait for laminar flow to properly establish. This is indicated by green lights on the display of the BSC. It is imperative that any foreign object, your arms included, be sprayed and wiped down prior to introduction into the BSC environment. After flow is established, spray 70% ethanol on the working surface of the BSC and wipe down thoroughly. All items, including serological pipettes and media bottles, should also be wiped down with 70% ethanol before placing them into the BSC. Cell culture flasks and dishes coming directly from the incubator may be wiped with a Kim wipe, pre-wetted with 70% ethanol. However, be careful not to get ethanol into the cultures as it will kill the cells. For example, watch out for the screw cap and membranes of flasks. Also, frequent movements in and out of the BSC will disrupt laminar flow. We are now ready for cell passaging. Assemble the 2 milliliter aspirating pipette onto the aspirator line. Be sure to turn the valve to turn on the flow. Open the T25 cell culture flask and tilt it back so that the medium flows towards the bottom and remove the old medium carefully by aspiration. Place the tip of the aspirating pipette towards the back corner of the flask to avoid scratching adhered cells off the surface. During this procedure, be careful to avoid touching outside surfaces. Quickly but carefully, rinse the cells by 3 milliliters of PBS. The PBS rinse will remove excess serum in the medium that would otherwise inhibit trypsin activity in the next step. It's important to add PBS slowly and towards the side of the flask to prevent cells from detaching. Swirl the flask gently and use the aspirating line and a fresh aspirating pipette to remove the PBS. Immediately after removal of PBS, add 2 milliliters of trypsin. Warm to 37 degrees C and incubate at 37 degrees for no longer than 5 minutes. After 5 minutes, check the flask under the microscope to confirm cell detachment. Return the flask to the BSC. Add 3 milliliters of complete media with serum to inhibit trypsin. Then transfer the whole mixture into a 15 milliliter centrifuge tube. Rinse the bottom of the flask a few times to remove most of the cells. After centrifugation, gently remove the tube to avoid disturbing the cell pellet and bring it back to the BSC. Don't forget to wipe down the surface of the tube with 70% ethanol prior to bringing it back in. You should be able to see a cell pellet at the bottom of the tube. Carefully aspirate supernatant while keeping the cell pellet intact. Resuspend the pellet with 3 milliliters of media, pipetting up and down until the solution is cloudy and the pellet disappears. Now it's time to count the cells. Transfer 100 microliters of cell suspension to a microcentrifuge tube, and load 10 microliters of cells from the microcentrifuge tube into each side of a clean hemocytometer. In the hemocytometer, each large square has an effective volume of 0.1 microliters. Count the number of cells in each area, A, B, C, and D. To avoid overestimating cell number, only count cells lying on 2 edges of each square area. For example, count cells that are overlapping only the top and right edges, but not the bottom and left edges. To calculate how many cells you have in your original solution, You must average the numbers of cells you count in all 4 areas. This number represents the number of cells you have in 0.1 microliters. To convert this number to cells per milliliter, you have to simply multiply by 10 to the 4th. Based on the cell density you obtained from the previous step, you will need to calculate the total volume of cell suspension you need to add to the new T25 flask to reach 700,000 cells. For example, assuming we have 3 milliliters of our original suspension, after counting, we get the following numbers in each quadrant: A 33, B 31, C 55, and D 21. Taking an average of each quadrant results in having 35 cells per 0.1 microliter. Adjusting this count to be in cells per milliliter results in 350,000 cells per mil. This means in our original tube, we actually have a total of 1.05 million cells. If the amount of cells we need is 700,000, we would simply take 2 milliliters as the calculations show. Always label your flask with your name, section, date, cell line, and passage number. Finally, place your flask in your designated area in the incubator. Double check that the cap of the flask is closed and avoid culture medium from contacting the cap while you are transferring. You're not finished, however, until you have finished cleaning your working area. For the purpose of this class, you need to dispose of any solid waste that has contacted cells or medium into the biohazardous waste container. Everything else can be discarded in a regular trash can. Before you close the sash, spray the bench with 70% ethanol and wipe down with a Kim wipe.